Okay, so for the next one, we have Anand Muthusamy. Can we have him on the screen, please? MITAV, we need them on the screen. There he is, Anand from Caltech. So Anand here is a powerful individual who is also yet another community organizer around olfaction things, such as Ricardo, myself, and others in this audience. So I think that there's a lot of us now, Anand, and that means good things are happening to olfaction. So he's going to tell you about engineering of binding proteins for continuous monitoring. And for those who don't know, continuous monitoring is a buzz phrase nowadays. It is very important for pharma, as well as other industries that have processes such as fermentation of biopharmaceuticals, which is a long process that if it breaks somewhere, you can lose hundreds of millions of dollars in 1 week. And yet if you stop the run early, you can save that money. It's also important for other industrial processes such as curing of biocomposites or composite materials, glues, industrial cooking, and many, many other things. Continuous monitoring is a buzz phrase that you will see a lot. It is $1 billion problem, at least for pharma, maybe more. And Anand here is one of the world's experts, so he will tell us all about it. Take it away, Anand.
Great. Thank you, Andreas. Can you hear me okay?
Yes, we can hear you and see you perfect.
Yeah. Well, thank you, Andreas, Carolina M, for organizing and having me here. Yeah, so I'll jump into it. I'm somewhere sitting in between Konstantinos and Lindsay. I really enjoyed both of your talks. I'm interested in the form factor of continuous monitoring on people. A lot of the sensors are general, Andreas mentioned, but I'm really interested in the relationship between people and devices that tell them more about themselves. Right now I'm wearing a continuous monitor to learn more about my blood glucose just for self-experimentation. But wouldn't it be nice if I didn't have to hook myself with that kind of device and, use a sensor in a friendlier form factor? So it's really nice to see this conference organized around that. And here I'm trying to take just a data maximalist approach. How many binding proteins can you make for how many molecules, even for scents we haven't yet created? Here's just a general scheme for an electronic nose. So that's just what we call devices that can pick up on scents and digitize them. And you may want to do this for threat detection or to learn more about your physiology as it evolves over time. And really what I want to focus on is the bioreceptor part. And the take-home message is we should think about bacterial proteins in addition to the obvious olfactory receptors and odorant binding proteins. If you want to create a digital nose, you need to have some receptor that's going to interact with the molecule in the air. And of course, the obvious go-tos are from olfaction, but I'll show a couple of schemes that show an analog between what insects and mammals have to do to process olfaction and what bacteria have to do for chemotaxis. And the reason we should care about that is bacteria have some of the most rigorous needs to evolve new interesting proteins, and time again we find interesting functions in bacteria. And compared to the hundreds of olfactory receptors, bacteria have tens of thousands of these periplasmic binding proteins, also known as solute binding proteins, that detect a variety of organic molecules. So why use sensors? Why not stick with just regular odorant receptors? There's plenty of work to do there. But on this plot on the left, which is from a nice recent paper, this group does a theoretical calculation of in entire chemical space, how many molecules could be odorants? Do they fit the chemical profile to be a volatile organic? And there's some 40 billion possible odorant molecules. And you may be interested in this because it could be one of those molecules could be a potential threat, or it could be a compound we just have not yet characterized. That is in nature. So from those hundreds of odorant receptors, they may or may not be tuned to one of these 40 billion compounds. Or if you just want to have a more powerful backend ML model, you may like to sprinkle in new receptors. So the gambit here is because we can make proteins with the tools of protein engineering, let's make a bunch of new odorant receptors and go to bacteria to find some nice starting points in addition to the mammalian systems. So this cartoon at the top just shows you the entire blue ocean is protein space in general. Native protein sequences are represented in this little corner here. So nature sampled some proteins, but definitely not all. And then we can push these blobs of native proteins using directed evolution, meaning we have an intended goal, we make a mutant in a protein, we see if it reaches our goal. If so, we keep iterating. And these are just various techniques, structure prediction to de novo design of how aggressively you design that protein to your own wishes. And the point is, this field is matured where we can design proteins from scratch. We can also borrow proteins from nature and crank on them to produce a function we care about, in this case, binding small organic volatiles. So sequence space is vast. Starting from scratch is hard, but fortunately, nature has given us a lot of starting points. Again, bacteria are wonderful protein engineers. And here's a scheme that you'll see that mirrors the olfaction scheme in insects. So this is in a bacterium. Most bacteria don't care about olfaction, but what they do care about is chemotaxis. They need to find their way in a complex environment and travel up gradients to find food. So if they find something they like, they want to move towards it, or if they find something aversive, they need to move away from it. And these periplasmic binding proteins, these Pac-Man shaped proteins or Venus flytraps that clamp on their ligand, bind all kinds of small organic molecules, ions and peptides, to bring those molecules to the ABC transporter, bring them into the cell, and that forms a signal transduction cascade. So the main takeaway here is there are tens of thousands of these binding proteins in bacteria working on chemotaxis, and they have several nice properties that you care about for continuous monitoring. They detect a lot of small molecules. They have large conformational changes, much more than most conformational switching proteins, so 10 to 40 angstrom movements, and they're reversible. More than that, various binding proteins have varying substrate scope. So some of them are very selective one-to-one. Others are pretty promiscuous. There are some PBPs that will bind any 5-mer peptide. So if you're interested in, you saw a nice talk about non-biomarker or biomarker agnostic, there are promiscuous PBPs that can bind an entire class of molecule. So that's the bacteria picture. Now, connecting back to olfaction, what you saw before is essentially turned on its side here, where there is a odorant binding protein in an insect, Sensilla, on the left here, zoomed in. These odorant binding proteins essentially serve the function in nature to move a insoluble or poorly soluble molecule from the outside towards an OR towards the inside of the cell. So at a high level, these proteins look similar and they perform similar functions. I think it's just an understated connection that indeed bacteria have an interest in chemotaxis and have similar proteins, just more numerous compared to what insects have evolved. So again, the gambit is to make new odorant binding proteins out of PBPs. And then I'll just leave with a few notes about how we engineer this. PBPs are nice in that the readout can be modular. We can couple them to gene circuits. We can couple them to optical readouts. These are nice for high throughput screening. So we can use bacteria as machines to make proteins evolve towards what we want them to do. And then once you evolve it, you can get rid of these tags and then immobilize that protein on an electrode like you would in an electronic nose. And this is what you really want for a device. So we use the green box for high throughput evolution, and then the aspiration is to immobilize these evolved proteins on an electronic nose. And then we know how the fluorescence mechanism works. Again, we want GFP readout because it's easy. So here's the PBP, it's binding its ligand, and this linker that we install, that you can install in any PBP, pulls out this glutamate. It essentially relieves the quenching of GFP. So this linker essentially is a way of communicating the binding event in a PBP, which you can't see, to GFP activation, which you can see. So this is a molecular mechanism and a general method of evolving or reading out the activity of PBPs, and it'd be generalizable to other conformational switching proteins. So the last thing I'll mention is we're able to take PBPs from nature and evolve them to bind things they've never seen before because they have some latent activity, as do many proteins. And that's the story of directed evolution. So we take a PBP that binds choline, 100 Daltons, highly hydrophilic, and then evolve it to eventually bind something that's 10 to the 8 times more hydrophobic compared to choline. We have a specific interest in neuroscience to look at neurotransmitters and drugs. That is not exactly relevant to olfaction, but the techniques of generalizing PBPs are. So the last thing I'll mention is, I'll leave this up for any questions, is, in neuroscience, there's a lot of interesting protein engineering work, but there's not a lot of cross communication with the olfaction field. So folks are really hyper focused on, taking a crude heat map like this, where you see a lot of general sensitivity to a bunch of compounds, and you want to really detect one thing like dopamine. But again, with the nice talk about detecting a variety of biomarkers or being agnostic, you can actually just save a lot of this work and take PBPs that are pretty promiscuous from nature and stick them in an electronic nose with many steps in between. So I just want to thank our variety of collaborators and, again, the organizers for having me. Happy to take any questions.
That was great. Thank you, Anand. So unless there's questions from the audience, I have one. And have you seen any, you showed something very compelling, which was that you can evolve a protein towards what you want it to bind. Mm-hmm. This has been the holy grail for many years now. And we've seen this work with GPCRs, rhodopsin. You can evolve it towards being sensitive to a different part of the wavelength. We've seen this with whole organisms such as algae that in my lab we've evolved to photosynthesize in the IR and in the ultraviolet. Now with your proteins that came from bacteria, do you see a way of maybe, there's two questions. Do you see a way of maybe evolving them to the point where they actually become GPCRs? Like keep challenging them with odorants until you find what biology found?
Yeah, that's a wonderful question. You have a good intuition, because better than that, some folks have found that PBPs have poor sequence homology, but extremely high structure homology with their GPCR counterparts. So several glutamate binding proteins essentially nearly overlap with the metabotropic glutamate receptor, that GPCR. So already at a structural level, both growth structure and binding pocket structure, there are PBP analogs of GPCRs. So the answer is nature's done it to some extent, and we could push it further. So yes.
This is Adam Feldman again. That was an outstanding presentation. And the thing I think of when you're talking about continuous monitoring and, monitoring of biologic processes, there are a number of things in medicine that, patients under anesthesia, patients in the intensive care unit who are undergoing a, septic episode, they have a systemic infection, or we are always trying to identify risk factors for developing sepsis after certain procedures and in certain clinical situations where, you could use something to predict or identify it before it becomes physiologically present. And I guess the question then is, how do you identify what your target's going to be? How do you know, that's the hard part is, right, you can develop the monitoring system, but what to monitor and how you combine those two things.
Yeah, so, the human metabolome database, which anyone can Google and access right now, has some 300,000-400,000 entries. And most of those markers show up in blood. Some of them show up in interstitial fluid. It's less well characterized for excretions and odorants. But I think Nick Law made this point about there is some mapping there, and that just has to be the task of the basic science is to find that mapping. For stuff in fluids, there are a few privileged molecules, like we know cortisol is interesting, glucose is interesting, and so on. So, so say for physicians, they have maybe an endocrinologist will have some hormones and glucose and insulin of interest. So there's some privileged molecules, but there are probably many in that entire human metabolome database that are just untapped or undiscovered. And then I don't know if you have folks from the Google Brain effort to map the volatilome, or I'm sure there are many efforts to map the volatilome. But essentially, as a protein engineer, I wait for folks on both ends, people doing volatilome mapping and physicians who have immediate needs to tell me what to make. So basically, there's some privileged molecules that we know from decades of biochemistry, and then also a lot of the PBPs are promiscuous enough to give you a pattern for any unknown sample. Same for the odorant receptors.
That's fantastic. We have to move to the next speaker, but Anand, I need to talk to you some more. And my next question, I'm preparing you now. Don't answer it here. Is about OBPs, odor-binding proteins, especially the ones from the pig and their pH sensors. Fantastic work by Anand. Thank you so much.